so unquestionable that even evidence that comes later through many researchers' works, they have to go through those restricted pipelines and they don't find other researchers' attention. So Professor Wadlakonda and I got together under this, like that. He also has challenges for so many classical metabolic pathways. So thank you, Professor Wadlakonda. Thank you so much.
Thank you. Thank you. Sorry, I couldn't be present yesterday.
Who is online?
Yeah.
He wants to ask something?
No.
Okay, fine, fantastic. So, hello, Federico. I just wanted to say hi if you're hearing this. Yeah. Then, can we go on to the next talk then? I would like to go on to the next part of the talk. That is.
Will you share the PowerPoint?
Yes. Free. So, thank you. Yes.
The previous slide there was a slide number. For me to identify some of the questions.
Yes.
Based on slides.
Now they are.
You want whatever you want will be done. You know, I have those PowerPoints separated also. I have them together also. Repeat. All right. So let's go to the next. We are already 7 minutes. Delay. So let's see the second part of my session. That is electrophysiology and erythrocytes. All right. After this, we'll have some time. In the next session, I've kept it lighter so that we can discuss, and I'll try to summate all that. So here, till now, what we did from chloroperoxidase, we jumped on to cytochrome P50. From there, we jumped on to respiration. Then from there, we jumped on to photosynthesis. Then from there, we jumped on to visual processing, and then neuronal relay, all that. Now we are coming to very, very complex ionic homeostasis, electrophysiology. Electrophysiology is also involved in neuronal function. So neuronal relay, that's what electrophysiology is, a very complex field. So from simple enzymology to bioenergetics to electrophysiology, that's what slowly building system to macromolecular. Professor Wadlakonda, what he asked is, how do you integrate those cellular systems and their connection with so many induction pathways, so many genetic controls and all that comes much later. Even now, we don't know, the chemistry part of it is also not resolved properly. You see, when the genes and the neuronal, hormonal, all those things come, of course, they are more important than this. But as a man with simple chemistry and physics approach, with hardly any resources for experimental biology, I like to stick to simpler systems. That's what I have explored. So now let us come to electrophysiology and erythrocytes. Okay, here these slides, I'm sorry, but they are very number intense. All right, here you have to be with me. I say some things, and you just see what I see. You see what you hear what I say, and try to go along with it. Okay. Now here is the approximate ion distribution in the red blood cell and in the serum. All right, it's given in millimolar. So in and out. So in classical electrophysiology, you always say in, out, right? In cell, outside the cell. Outside is serum, plasma, whatever. So you see that potassium is 139 millimolar, whereas sodium is 150. Let's not see all these. The calcium is very low, magnesium is significantly lower than sodium also, and then chloride is low inside, chloride is high outside. The thing is, between potassium and sodium, they are both group 1, right? Metals, right? One electron outside the last shell, which easily can be knocked off. It can give out. So that's, and they are very similar. How is it that cells have high level of approximately 140, the same outside, whereas the outside potassium is much lower, sodium is still higher. So how do we have this? When particularly you see the solubility of these things, you have sodium chloride solubility is much significantly higher than potassium chloride. Now they have these are the abundance in terms of their availability in on Earth's crust. Sodium is higher than potassium. What is the logic? See, sodium is of a lower size. So if you would imagine a molecular sieve to be functioning on the cell membrane, right? Now you imagine which one can you sieve in? You can sieve in the smaller or the larger? Small, correct. So you would imagine if you have a larger sieve, the larger one and the smaller one will also go. You can only sieve in a smaller one, but that's not what we see. We have the larger one that is inside. So how is this? This is one of the most complex problems that ever man faced. I like to think like, because it's a very simple thing. But the answer to that, there are so many characteristics that are listed here. We'll come back to this if time permits. Maintaining concentration of and see these ions and metabolites is maintaining their concentration is called homeostasis, right? So how does that do? We are not looking at homeostasis at a organ level or at a tissue level, please. I'm looking at a very simple cellular system in a very non-sophisticated modality. Now here, why K is more than Na in cells? That's the fundamental question. Okay. So I've changed the slide order based on Professor Sharad Bhartiya's recommendation. That one I couldn't, but this one I did. So now here I directly introduce the other theories involved, and then I'll go to challenging the established idea. So here, cell membrane outside in cell, ideal electroneutral state. You have minus 33 plus plus. Now, if there is to be a disparity in charge or potential thereof, here there are three schools of thought traditionally. All right. The third school is my own Murburn school, which started only recently. For more than a century, there has been a fierce debate going on. One is called membrane pump theorists. The other is called the association induction hypothesis or sorption theorists. I don't know why your friend is not here. So he was asking this question. I said I'll talk about this. So now you see the association induction. They believe that the transmembrane potential difference and the fluctuations thereof arise due to fundamental process of adsorption. The AIX theory is based on sorption of ions and other molecules onto surfaces of proteins, membranes, anything, right? So this is one theory, and this theory says that solve active pumping of ions across spaces. My theory says that there is electron charge separation. It's not just an ionic theory. Did you understand what the difference between the three theories are? Here, there is electroneutral. Basically, here what I'm saying is, you see, minus, minus, plus, 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 minus. There is an extra charge created here, and the other charge is mobilized. Did you understand what I'm saying? Here, the sodium or potassium is either adsorbed or pumped. Here, maybe superoxide is produced, something like that, right? Right? Did you get it?
What does absorb mean?
Adsorbed.
What does that mean?
It's a physical process of, now you see you go to some room, it might smell, there might be some gas, and then once the gas goes off, yet the surfaces would retain that aroma, for example. That's because of adsorption. On the surface, it can just smell. There's no chemical reaction per se, but then some due to physical characteristics, they adsorb. It's not absorb on surface, right?
So the mobility comes down, is it?
Yes,
What
yes,
happens in
yeah,
that?
yes. They're not available for bulk interaction. Yeah. So now here you see that's what. So here in the membrane pump theory, membrane is the role. It's what is functional. It regulates what comes in, what goes out. It's like you have a passport, the passport officer sitting at the airport determines whether you come in or go. So as per the membrane theory, all that reside in India are determined by the passport office. All right. And as per this theory, I'm just trying to give an analogy. The bulk phase determines what stays in. Bulk phase. You see the difference. The bulk phase in and out determines. Here, the membrane determines. Here, I say all of them. It cannot be, if you want to measure the transmembrane potential, it has to be a thermodynamic interaction between the two phases mediated by the interface. It cannot, basic, all of them have to interact, and it's a dynamic process. You cannot just say one portion does or this portion does or that portion does. So having introduced the different schools of thought in this field, I am going to the next status appraisal of this is the updated perception difference between the three schools of thought. I'm not going to go into the detail. Basically, if you see, it's there in my JCP paper. So Austin has no role in both schools. They have strong disagreement between each other. They don't see eye to eye, right? The two schools. The association election is primarily physicists and physiologists. This is hardcore physicians and biologists. Generally, I mean, there is no such hard and fast rule. But then, if I would analyze the background of the people, it's like that. Now, here I would like to consider myself somebody with some chemical leaning more. So there's a physics, chemistry, biology, that kind of distinction. So yeah, there is there any, so they have dissimilarities and similarities, and this school of thought has also got similarities and dissimilarities. So in this paper, the last paper that the visual construction paper, I said that it is high time that rather than seeing only select set of observations and accepting them as to be the only things that are operative, we should be able to explain based on the large diversity of observations that have, and we should have a tangible model that can explain every single observation rather than I take one set of observations and say that is what I explain, right? You should have a theory that accounts for everything, not just accounts for whatever you choose. So let's see acclaimed perspective. This is very important. I hope we would have had more students here. But then the point is classical. Now here is where you see as a scientist how you accept theories. The classical model would require you to have these things in the cell. First, the cell should be able to, if it has to homeostasize, if it has to maintain its volume, content of the ions, metabolites, and all that, it should be able to measure things in and out, particularly in. All right. Then it should be able to recognize, specifically identify if it has to transport. And the transportation or translocation is based on pumps, porters, and pores. Pores are also called channels, right? Right. So now, for the pumping in or outward pumping, you need ATP. For the porters, there are uniporters, one direction, antiporters, one transported this way, another transported that way, or then symporters, two are transported anywhere. And then pores, channels that get operated by voltage based on some ligand binding, and then even tension, pressure, things like that, right? Can also supposed to open channels. So the classical perceptions are vitally deterministic, assuming an intelligent brain. If you think this is something else, you think if a membrane were to determine what comes in, what goes out, it would need all these attributes. do you think that a membrane is capable of all this, including its protein? How can it network, maintain its intelligence? That's something that I doubt. All right. So here, the transmembrane potential, for biologists, uh, this would have been helpful. So how is the transmembrane potential varying based on ionic contents? That's what I've drawn here. So transmembrane is deemed to be critical and is determined by regulating the various cations primarily. All right. And so here, uh, the TMP supposedly results out of the net differences in cationic potentials in and out. So if you see here, I've shown two systems. One is a squid axon, which is the most popular model to study nerve function, because contrary to small nerves that we have, the squid axons are huge, long. So it's very good experimental model. Right? So that's why they use the squid axon. So that's what I've shown here. So you see, basically, these are the diverse values. Actually, we should be able to sit and see in detail and think about it so that you will understand the issues involved. Now here, let's start to understand what ion pumping entails. All right, ion pumping entails that, suppose if there is no channel, we would, suppose this is in, out, we'll try to understand something by simple water pumping analogy, just like we did for photosynthesis yesterday. The current flow, we try to consider it with water. Here, same pumping also we'll do like that. If there is no channel, what will happen is, if they are of the same level, if water gets pumped out, slowly this level will go down and this level will go up. You agree with that, right? So now, but then that doesn't happen. So now, how are you explaining the physiology is, you have pumps that pump out as well as channels that allow in and out motion. That's what is happening. So basically, internal K concentration is 10 times Na, whereas exterior has 40x concentration to K. Bidirectional active pumps are and passive channels explain cellular physiology. This is the classical model. Bidirectional active pumps and bidirectional channels. All right. So here I will try to break this concept. The classical, one of the best known members of this is called sodium potassium ATPase, or an ion pump. Okay, it's an electrogenic ion pump. This is the crude structure wherein there is a beta part protein separate, and there's an alpha protein which is something like a Y, like a pump. You have a pump, right? Hand pump, that kind of a structure, but the pumping handle is something here. That's why it's been called a motor protein or pumping protein. So here, what happens is, it's supposed to, one of the most famous tussle in this field was Freeman Cope and Glynn. The very famous paper is in Trends paper. It's a Trends paper. So they had this tussle where this association induction hypothesis, the sorption based model, people fought with the membrane pump. Membrane pump are basically British physiologists, right? So British physiologists are Mitchell, and also those all people who advocated membrane pumps. So here is the sodium, 3 sodium ions first, then they're supposed to bind, and then ATP is supposed to be converted to ADP, get pumped out. At another temporal phase, then 2 potassium from outside, comes in. I've drawn this as a separate one because this is of a different conformation. How the classical theory explains is, at one configuration, it has a higher affinity for sodium. At another configuration or at another conformation, it has higher affinity for potassium. That's how it is explained. Okay, so sadly, Freeman Cope committed suicide, and this thing was also settled. Purportedly, England claimed that Jensku had isolated this protein which pumped all this in a definite deterministic ratio. So if you see the classical model, it is something like this. This is the resting state. Now ATP binds, then 3Na comes. You see, what does it entail? Now Na goes like this, gets bound, then the channel should close, right? And then you should have this channel opening, hydrolysis, and this gets pushed out. That's what is called pumping. Then this proton should come and block this. And then what is happening is it's like working like a very systematic, sophisticated, very highly intelligent molecule that is capable of binding definite number of ions, identifying them, and then systematically one channel opens, another channel closes, another channel opens, and another channel closes, like that across the membrane. That's what it's doing. I'm not going to get into details, but the standard textbook shows there are at least 15 steps. Here, I have modeled it with 12 steps, basically. If you just consider basic 12 steps, actually, the more the number of steps, do you think it's going to be easy or difficult in terms of working? Yeah, Chinmay? It's more, the more it is, it's more difficult, right? That many steps have to be systematically, repeatedly done. One goes wrong, the whole thing goes off. So I just considered 12, okay? Now critiquing the classical view. At the outset, let me ask a few questions. Deterministic quantification and regulation of hundreds of molecules and decades of ions. How many different ions you have? How many different molecules you have? Do you think all these molecules and all these ions can be controlled by channels, pumps, and all that? Is that possible? In that case, if that is the case, then if all these are controlled by sodium, potassium pumps and all that, why, if you have hypertension, why should you not consume sodium? Even sodium, potassium ATPase is there, it will only pump in a deterministic manner. If it is not subject to the laws of physics, if the thermodynamic equilibriums don't operate, and if this is what deterministic pumping is, what is operational, and how many proteins do you think, where and all would you place this protein? Imagine a protein like this. Now you have, how many would you want to place? X number of Na, X number of K, then X number of chloride, all, how, where, how? All that. That is one question. And they have to be, remember, for them to work, they have to know the concentration inside and outside. They have to measure, right? Yeah. So even if specific channels and receptors were present in all, their surface area would be much lesser than the total surface area. Then selective ion sorption at membrane interface of proteins, the ordering of water in cells are not considered in the classical treatments. Yeah. No reason provided why ion permeability changes during various phases of action potential. I said that in the last talk, right? Yeah. And then you have excitability and conduction of neurons could be afforded by several inorganic, and once again, Farachi's work, Farachi's writings, and Tasaki's and Terakawa's works mentioned in the earlier talk. I did that. All these things you can have other, suppose quaternary ions can induce the same kind of effect. How could you explain that if it is via specific ion binding to specific channels and specific proteins? You imagine some kind of an interactive equilibrium and thermodynamic principle, then you can explain that. All right. So here, I would like you to indulge your critical faculty like a simple school-going student. Just like Hans Christian Andersen's, that, you know that story, Emperor's new clothes, yeah. You know that story? Now I want you to be like that small kid. Now you see, Cells are 2 to 2000 microns in dimensions generally, right? So surface area to volume ratio of a regular 3-dimensional cubic hexaedron cell will be 3:1 for the smaller cell and 3:1000 for the larger cell. How do you explain the deterministic pumping in these different ratios for the same type of? Now you have a macrophage, right? Macrophage changes its structure. Have you seen a video of a macrophage going through like that? It changes topology. Mass transfer is dependent on what? Surface area and the gradient, right? So even if I don't understand how for varying regular geometries, for example, tetrahedron, hexahedron, the surface area to volume ratio varies in this manner. Conversely, for a given unit surface area, the volume changes like this. How can the stoichiometric activity of sodium potassium ATPase, which I will call NK, result in the desired outcome within all these cases? How can NK sense the cellular geometry to cater to a vitally deterministic mandate? That's the question. Did you get the question? Yeah, that's a critical question. Now then, let's see what stoichiometric electrogenic pump simulation gives us. Simple simulation. Let's not go for complex simulation. All right. What is an electrogenic pump? Why? Because it pumps 3 sodium out and 2 potassium in. So you have a gain of charge, ion charge, right? So that's why it's working like that. So now imagine, now you start with a simple system where you have same volume inside versus covering has also got same value. Now you have similar pump, the number of ions, say sodium, potassium, you start from a same volume, same amount of ions inside out. You try to get it to pump without any channels. You try to pump. What will happen? You will invariably end up with all the sodium going outside and all the potassium coming inside. Yes or no? Because 3:2. So theoretically speaking, from an initial state where there is equal sodium and potassium, this electrogenic state pumping cannot explain anything, right? Now, if you so for other geometry aspects it cannot, from any initial concentration it cannot, and then for very realistic stoichiometric pumping it cannot. Now the point is, how can it work? Basically, is that pump it means that, see, if Na and K pumping ratio is 3:2, it means that the ratio of total quantity of Na plus crossing the sodium channel, now we are including the channel, right, from the exterior to the cell interior, and the total quantity of K plus crossing the potassium channel from the cell interior to cell exterior is 3:2. That is, this is such a mirage that was created. You must imagine. Now, exactly the amount of sodium that is pumped out, if it has to homeostasize, that means that the passive channels should actually counter the exact effect. So is it explaining anything? You ask yourself, is it explaining anything? How are we all taken for a ride with this? Did you understand my point? Yeah, okay, now, so now there is a membrane, right? Now you don't first imagine that there are any channels. Now, you just imagine there are pumps. In that case, can it justify anything? No, because it will all lead to anything. You start from any initial state, it will not give us homeostasis or the final steady state, right? Now you have channels and pumps. Final, whatever we have the in our functional state, right? In the physiological, the concentration that is.
Charge is balanced.
No, I'm not talking about charge balancing. I'm telling you about the concentration differential that exists. I showed you some concentration in the cellular versus serums differential. I showed you, right? That's what I'm talking about. Now you hear, what you do is you have to understand a very simple mirage that was created. Now, if X number of sodium potassium channels are present, or not channel, pumps are present. Now they are supposed to pump. All right. Now When can they give a pumping that is viable? When all the number of passive channels that exactly counter the effect of this pumping activity. So it doesn't explain anything. When can this otherwise conversely stated, it means that the passive active and channel. So it is neither explaining the initial state, nor explaining the steady state, nor explaining any pumping, nothing it does. Okay, I'll tell you one thing. It's like this. One guy goes to a fruit store. He says, can I, how much is this banana? He picks up some bananas, right? And he says, oh, he picks up a bunch of bananas. It's, sir, 30 rupees. I'm fine. So with the other hand, he picks up 2 oranges. He says, how much is this? Ah, it's also 30 rupees, sir, oranges. So he keeps the banana down. With the other that hand, he picks up one apple. He says, sir, 30 rupees. So he keeps this down and he starts eating. Now, the point is this, he doesn't pay. It's supposed to, the paying is supposed to account for everything. It is supposed to reason that there's a transaction, something like that. Now, what is this theory supposed to explain? You tell me, what is the pumping, all that supposed to explain? It's supposed to explain the physiological concentration of ions, sodium, potassium difference. How can it explain? It must explain from a steady state, I mean, from any state to any state, it should explain. Now, with this simple thing, what I'm trying to tell you is that it was a mirage created wherein the channels are supposed to be countered by the pumps. And the pumps are supposed to be countered by the channels. How can they be? Ask yourself. Did you understand what I'm saying? Did you understand? Basically, pumps are passive. And channels are passive. Pumps are active. Is there any connection between them? No. Is there any geometry that binds them? No. Is there any intelligence that binds them? No. One is active, one is passive. How does the active know what is the passive doing? How does the passive know how the active one doing? How do they know how many are there? How many? Where are they located? There is nothing to control anything.
Passive doesn't need control. No, it's osmosis driven. It's osmosis driven.
Yes. So why did the passive work? Passive works because the active works. How does the active know what to? See, the point is, it was created. The mirage was created, wherein it was supposed to explain what, but from any state to any state, it does not explain because there is no logic to connect one, and there is no.
My question is, does passive really need to know? Because all it cares about is the concentration gradient, since it's osmosis.
So now I see you're right that way. Now, if you take suppose now let's say 50-50 inside, okay, of sodium and potassium. Now you pump 3 sodium out and
Dr. Murali, Dr. Murali, I would like to actually have a comment on this particular thing. Okay.
Yes. Can we have later, because
just 2 minutes, just one and a half minute, not more than that thing. Let us look into sodium potassium distributions. Sodium potassium distribution within the cells. First thing is sodium influx initiates actually the metabolic pathway. That is number one. Number two is a differentiated cell is rich in potassium inside. And when it comes to your action potential system, it is a cyclic change of sodium and potassium turnover. Potassium held by differentiated cell is thrown out in response to what is called activation potential, and that activation potential is activated by ATP. ATP is actually participating in this particular system. Under that particular condition, what happens is, whenever sodium is entering, it is also thrown out immediately, simultaneously. Therefore, there is a cyclic exchange of sodium and potassium. As the wave propagates, this particular cyclic exchange returns back to normalcy. That is the fundamental theory of action potential.
All right. Can we? I'll come to this later, Professor Varlagonda. Please bear with
Okay.
me. Yeah.
Okay.
Thank you so much. Yeah.
Right.
Thank you so much. The point is, Professor Vandra's question. I think I'll show you with simple modeling number later in the paper that I have. We will talk about it. Can I just move on? Yeah, no problem. Thank you so much. So this clearly shows it was just a mirage and does not explain a thing. And I'm very confident when I'm saying that, because it's a very simple thing. It's a logical thing that was missed out even in Peter Mitchell's theory. See, you are pumping out and it is coming back in, pumping out, coming back in. There is no real gradient ever existing. There is nothing. It doesn't explain. That was a mirage there also. This pumping theory is a mirage. It doesn't really explain anything. You imagine how can the work be done like that? We'll come to that later. Anyway, the second part of it is the ions that are supposed to bind. I would have bought it if it was different sites. The same sites are supposed to bind the sodium and potassium. Now, because it is bound separate, it goes out. What is it that prevents the sodium to come and bind back? For that, it is assumed that one state has sodium preference. But then I don't bind. Did you understand my statement this one? Now it pumped out sodium, right? And then it has to open up, right? And then it has to take in potassium, for example. But then there is enough sodium also there. In fact, more sodium than potassium there. Why should it bind to potassium? Because the sites are same, also same. There is some need to be some different sites to be exposed or something like that. Nothing like that is known. So then the accounting the theory, there are the classical treatment shows the chemical gradient to be based like this, electrogenic gradient to be accounted by this. These are the explanation terms. I'm little lagging behind. Let's go fast. So here you see the calculation for the simple thing. This is done in so many physiology labs. All that you have to do is put a realistic condition where the ADP and phosphate ratio to ATP is say 0.1 or something like that. Okay. Here you have the overall. It's not negative. It's positive. And you can even do a single molecule simulation wherein you can take a simple say 100 nanometer box and try to put some ATP molecules in that, but at a low ratio. It will not pump out. So basically, for the thermodynamic feasibility itself, you need high amount of ATP for it to be viable. So it can be only when these things have high ATP. So if sodium or potassium or ionic transmembrane potential, all these were affecting the ATP synthesis per se. How do you explain all this? See, as far as this is concerned, it is supposed to bind ATP and pump it, right? That's the deterministic mandate. There is no other thing. And this equation is supposed to account for it. But then, how does it? Then assessment of probability. I have just taken 12 steps. And 1 by 10 is if there are totally 10 ions outside, if your sodium ion is 1, I've given it 1. So, and binding dissociation, that process I've given half. So if you make a serial calculation, so for non-selective and reversible process, it's very low. But for selective, even if you give that sodium and potassium affinity changes in different ATP bound and non-ATP bound. Okay, because ATP is binding where? You see, ATP is binding here, somewhere here. How does the affinity change here? It's on a different region. So I don't know. So even
That could be because of the conformation change of the.
Yeah, conformation change. How does conformation change the affinity? See, this is something that has been given to us. There should be a reason why sodium and potassium. We know that proteins, preferably the carboxylates, we know the series, it prefers the potassium over sodium. But why sodium over potassium? It doesn't explain. So here, even if you account for a selectivity in the process, it would still be a very low probability process. This 13-step process, pumping process. And then we have, even without the pumping and channels, protein, this sphere, there's a very famous experiment. It gets the same transmembrane potential, the active, what is it called? Action potential. Action potential signature. Even without channels and pumps. All right. So here, compilation of ion distribution data. Brindley had compiled the ion distribution in a vast different number of cellular systems, and they don't buy the transmembrane potential purely don't agree with the ion distribution alone. All right. So also plants don't have NK, sodium, potassium, ATPase pump. They show this. All right. So Murburn purview. Now, what I say is that there are multiple aspects involved. Purely, there I do not negate that there is some specific adsorption, some specific transmembrane transport. I do not negate that. What I'm saying is, you look at the co-solvation of the iron along with the anion. People have left this picture. You see respiratory anions, carbonate, bicarbonate, acetate, phosphate. You see their solubility. For example, carbonate 1.7 into 10 to the power 3 potassium versus sodium 4.4 into 10 to the power 2. Calcium very low. You see acetate 6.9, whereas chlorine is the other way around. So if you imagine a status where chloride is outside and phosphate is more inside, which one would be preferable inside? Phosphate you see here, 10 to the power 6, 10 to the power 5. Did you understand what I'm trying to say here? See, you have a membrane phospholipid here. It's full of phosphates on the surface. It's a phospholipid, right? Negative charges. So now you have an electrostatic repulsion. Phosphates are retained. Now you have some kind of a thermodynamic equilibration where the membrane protein sodium ATPase just ATP binds and hydrolyzes. The phosphate would prefer to solubilize, co-solubilize with what? Potassium or sodium? I said there is a thermodynamic propensity, delta G of formation, solubilization, all that. There is in physics itself, the physical milieu itself, there is a selectivity. You see that? Did you get it? What I'm saying here? If you see chloride, sodium is high, potassium is low. Just see, don't go into the calcium and magnesium. Just see potassium and sodium. See, potassium phosphate is much more soluble than sodium phosphate. Selectivity is lies there. Did you understand? Not in the selective binding and pumping. Primarily there. And then the second part is there is a significant aspect called, I did not put those equations, but we'll come to that. When we come to the equation, I'll go there directly. Okay. Here you see here these reactions. These redox reactions are also very important. I should have shown those equations. I did not put those, but that's okay. Here you can get it. You will get it. Here you see these values of redox reaction, ability to get reduced and oxidized. You see potassium in getting reduced 19.3 kilojoules per mole. sodium minus 2.1. You see there's a difference. You see there's a difference. One is negative and one is positive. Yes? You see that? Now you come to here, this slide again. Now you imagine this is a respiring cell. This is an alive cell. You have an oxygen-centered higher amount of superoxide on this side and oxygen on this side. Yes, you can imagine that scenario, right? So that will necessarily also enable potassium coming here. It's a thermodynamic equilibration. It is not necessarily a selective ion pumping. The vast major activity. Here, the physical ambience itself is such that it does. So there is, what I say is, I'm not 100% sure how it works, but definitely not by that way, because it doesn't explain anything. But this could be when NK hydrolyzes, it recruits there is also ROS here that enables the potassium to get co-solubilized with the phosphate that comes out here. Rather than multiple channels opening and closing, potassium phosphate gets here, sodium goes out by some other way throughout. Now, this is the basic rule. See, phosphate does not go out because it's a phospholipid. Now you know why phospholipids are phospholipids on the membrane. Right? For electrostatic repulsion, right? Phosphate, you don't need that much phosphorus. Our system retains all the other things we have exchanged. That's the rationale. Now here you see, how do I justify the new theory? This is the sodium potassium. See, it's not like it's porous, rather it's an open kind of a molecule. And this is the alpha, this is the beta. It's like the Portuguese, the hair, the tail on top, beta, one over. You look at the underside of that beta. They don't know what is the function of, the classical model does not espouse any function for the beta monomer. Now, if you see the beta monomer, underside the beta monomer, there's a large amount of lysine and arginine. Lysine and arginine. So what does that lysine and arginine do? Why? What is lysine and arginine? Positively charged. What does positively charged thing do? It recruits negatively charged species. What is a negatively charged species that you will expect in the membrane? That is mobile. Superoxide. Oxygen and oxygen-centered radicals are 5 to 6 times more soluble in lipid phase than they are in water. This is the reason why we have a lipid membrane. This is the reason why we have phospholipids. Did you understand where I'm going to? Now, are you able to connect the dots here? No? No? Oh, okay. See, here phosphate. Cell has respiratory phosphates.
Yeah, about
Okay.
the structural part.
Structural part is a beautiful part here. There is no role whatsoever accorded to the beta top part. Now, there are sodium and potassium here and there, right? What does this do? It recruits superoxide around that region. It's a stochastic process. Now, if you have superoxide there, it will have equilibrium, right? This is not a stoichiometric process. What it will do is, at this membrane, it will preferably like to push potassium in, sodium out. The equilibrium in the presence of superoxide, it will be like that because the thermodynamic mandate is like that. Thermodynamic mandate is that.
The point which I'm confused about the structural part at least.
Yeah, so
You can go back to the structure there.
you did not get how the structure helps that mandate.
Exactly.
Yeah, I got it. So now here, I got it. That's what I'm saying. Here, you want this area to be doing something, right? Yes. What does it do?
Here you see this beta and alpha who are interacting with you, right?
Yes.
Beta subunit and alpha subunit is interacting with each other.
Yes.
That case is saying that the lower part of beta has more of lysine and arginine residue.
Yes.
Right?
Yeah.
What about the part which is what about the part of alpha subunit that is interacting with the beta subunit?
It is this open drum-like thing. It sits hovers on that drum. No,
It's not completely sitting on the
it is sitting, but it is not like sealed tight and all.
That's fine.
Yeah.
In that case, if there has to be complementary negatively charged residues on the alkylics, right? So
No.
the alpha subunit.
There is what I'm saying is here. This is not really completely fitting like a lid and something like that. It is okay, something like that. So there is a breathing in and out. It can go.
I'm really sorry.
No problem. Even if one person is interested, somebody like you, I want to. Next, I realize that the next part is.
I dive in for 1 minute here again. Well, although sodium potassium exchanges are, in case of action potential, are picturized as individual sodium potassium exchanges, they are not just sodium potassium exchanges, they are also associated with phosphate and glucose exchanges. Taking at least from renal cell systems, proximal tubules, where actually sodium potassium exchange is also associated with glucose and also phosphate. In most of the cases, there is what is called a phosphate sodium exchange, and potassium retained inside the cell, in the differentiated cell, gets out in all the activated cells where ATP production is activated by RBC. That's my assessment.
Yeah. The point here we are discussing is the structural correlation with the thermodynamic function.
That is exactly what exactly is happening is interacting molecules are not taken into consideration when you are only explaining sodium potassium exchange. There are also other molecules, neutral molecules, phosphate molecules, which are also involved in that thing. I think you have what is called fructose dependent systems, which have a different role when compared to the glucose system. Fructose dependent systems will inhibit glucose uptake here. But in case of differentiated cells, glucose uptake is not hampered by fructose and sodium systems.
So Wadlakonda, here we are discussing structural correlation with thermodynamic activity and the immediate function here. That's his question is how do
am I to consider that it is independent of other molecular alterations that are taking place in simultaneous?
Professor Wadlakonda, we are not going into the complex network aspect here. We are just
Okay. Go ahead. But there are other dimensions that need to be taken into consideration when you are actually looking into this particular structure. Is my highlight. It's okay. Fine. Go ahead with your thing.
Thank you so much. See, the point that I am trying to say is what happens is a thermodynamic equilibrium. All right. And the structure enables that. Now, you suppose if the lipid membrane did not have this kind of an opening and it's a regulated opening, you would agree? All right. It's a structure. Oh yeah, you are a structural biologist. Correct. You take your time. I forgot. So the point is this protein has got lots of cysteine all around. All right. And then these are the different views. I think you should take this underside view, all that. I thought to me it was very simple. Then there is phosphorylation of NKA, Dr. Pratik yesterday. The ATP that is bound here, the structural change that is supposed to result in the conformation change and the affinity change of sodium and potassium. It is supposed to be a phosphate, the Y part bottom bump. Here, the ATP is supposed to get bound and the phosphorylation is supposed to happen. The phosphorylation happens at a distinct space compared to the ATP binding. So how is that? And then this is supposed to make the arm swing and then do some action, then all that kind of in a repeated, systematic, deterministic manner. How is the phosphate getting phosphorylated at a very distant place compared to the ATP binding site? So this is the conformation superposed, the two different ATP bound phosphorylated after the phosphorylation one, E1 and E2 states. That's what is shown here. You see, there are so many phosphorylations that occur on this protein which are not accessible by kinases. Right? Did you see what I'm saying here? This is a transmembrane region almost. Kinases cannot access that. So how do you explain these phosphorylations? Then so there are 7 phosphorylation sites reported, some not reachable by kinases. The phosphorylation site of D369 is relatively distant from the ATP binding site. So involvement of ROS can explain this. This is a redox protein that does not have a redox center in the sense. There are lots of cysteines, I would not call them redox centers, though they are redox centers, but classically, a flavin or a heme would be called a redox center, basically. So here, phosphorylation of NK, and you see the structural diversity of the number of NK modulators. What does what? Yeah.
That whatever you're saying that earlier, whatever all the proposals and all the structures and things, everything is same. Just the difference is that the reactivity.
Not everything is same. There's a significant difference. It is not a deterministic pumping. It is not a stoichiometric pumping. It is not an electrogenic
So
pumping.
you have probably theory that defines how exactly the proton, not proton channel, the sodium potassium channel function.
It's not a channel. Say it's a way of how do you call that? Suppose now you're going out, coming in, for example, you have entered some malls, right? There is a air or something like that from top, so that you there is a prevention of bacteria from outside coming in or something like that. There is
Dr.
when
Dr. Dr. Murali, one question, simplistic question. Supposing
yes.
there is a cyclic change of sodium potassium and cyclic change of phosphorylation and dephosphorylation of ATP associated with these exchanges, does it address your bioenergetic theory?
You are coming to the systemic thing. It does. Glucose
Yeah.
leads to the phosphorylation cycle, and it does lead to sodium potassium.
I am leaving glucose. I'll come to that thing later, but let us only concentrate on sodium potassium exchange with phosphate exchange, right?
Yes.
Supposing there is a cyclic change of sodium and potassium, which is actually propagated through the length of the neuron, simultaneously there is ATP formation and degradation simultaneously taking place, where phosphate influx and efflux also make an important role. Can we combine that?
What I'm saying is, here in this particular context, the structural biologist Chinmay and his friend, what's your name?
No, that is fine. The structure is a dynamic system. It is not just a static system, right?
Yes,
Living system is a dynamic system. It is not a static system. In that particular case, you have a system where cyclically ATP is broken down and reformed. Simultaneously, there is an exchange of sodium and potassium across the membranes, and differentiated functions are associated with potassium inside.
Yeah, rather than having a pumping action, we have an ion differentiation. That's the word I use.
Yeah.
The ion differentiation is not by virtue of affinity or all that. It's because of the thermodynamic property. Because the equilibrium stable. I just go on. I'll finish my slide. I'm already 5 minutes late. So what you see here, now imagine that sodium potassium ATPase is a redox protein. All right. It recruits, it has superoxide functionality recruiting. Now, if you imagine that, you can imagine how all these diverse molecules can affect. This is the literature. I spoke the literature. How else can you explain the ability of sodium potassium ATPase to be affected? These are all interfacial redox active molecules. So this is one explanation. These are not, they can bind anywhere in the interface and the protein. What one say is, we can I শেষ করে দিচ্ছি, please, I'm just so we will discuss this after this. So miscellaneous simple NK is just oxygen sensitivity. Uncouplers like dinitrophenol fatty acids affect NK. You see this fatty acids, dinitrophenol, there is dinitrophenol. Earlier, same thing was supposed to be active in mitochondrial membrane also. It is supposed to shunt protons here and there. How can this shunt? This is I've got say this is say 6 or some nanometer. Say some the dimension of this is approximately 5 times lower than the dimension of the membrane. It has to deterministically travel. All these, they're supposed to be some kind of no. This nitrophenol works across all membrane, all physiologies, because these are ROS sensitive molecules. And ROS sensitive oxygen is superoxide is also connected with the proton equilibriums. That's why we have this. So the next one is observation of ATP synthesis by NK. How do we have ATP synthesis by NK, which was quoted by Freeman Cope as the Ian Glynn as the anti argument to Freeman Cope's absorption thesis. This is the reason. You have ROS recruitant. ROS will always make ATP around, so that will then transmembrane peptide. This is a very strong inhibitor of NAK ATPase. You see, lots of serines, they can interact and then lysine histidine. Same, ROS active. Same, if you remember my talk, cytochrome P50 had a inhibition with N-term segment that inhibited cytochrome P50, and also had a lysine there. Same thing. You need to have the thing at the right position. So the effect of lithium, this explains the effect of lithium. You see, lithium is given to soothe nerves, right? Some neurologically disturbed patients. Now you see the dimension and the thermodynamics. You see the value much higher, and that replaces the equilibrium. This is what, and this is small dimension. See this. High value here. So the isotope redox properties determine what ions get to the nerves. That's more important. All right. So here, cardiotonic steroids. These are once again very important heart medicines. They are one of the oldest known medicines. So they are structured like this. You know, the frog toad toxin. You see they use in the darts and all that bufalin, that one. This is digoxin, obane, very important membrane study proteins. Now you see the classical theory says that they bind like this. Yeah, here they bind here, and here is where it affects that. Okay, now it is very specific binding. That is what it says. Now, if that were be the case, these are the structures of some of the. Then in that case, these are also structurally similar. Cholesterol, testosterone, cortisol, very similar. So then the active moiety is the lactone ring here, because that is the one that makes the ROS plus proton dynamic. So you see, even these molecules which don't have the steroidal ring, they are also effective in inhibiting. So in the next, I'm already late, it's already gone. So I have to finish this part quickly. Please bear with me. Hemoglobin, RBC, the traditional function is to transfer oxygen, right? Here, you just see the glycolysis pathway. You see the amount of the components that are remaining. The bisphosphoglycerate component, if you add everything total, if you see how much concentration you can get, practically it cannot explain the millimolar levels of ATP that you find in RBC. So how is RBC sustaining for 120 days without a nucleus and without a mitochondria? How? We do not have a viable explanation in the classical paradigm. That's what the next thing is. What I am trying to propose is, and I have demonstrated in my paper, is that iron 3 methemoglobin, iron 2 hemoglobin, and oxyhemoglobin. So iron 2 binds to oxygen is the same as iron 3 binds to superoxide. That's what here it is. So this can dissociate. This is the redox equilibrium here. When hemoglobin dissociates as iron 3 into the met form, you get ROS, superoxide. Then that can make ATP. That's the simple theory. So here it is. You see the beta monomer. You have 2 alpha and 2 beta coming together to make hemoglobin tetramer. The beta monomer has got an opening. This is the one side, this is the other side. There's a small opening here. Why is that opening important? Now you have that bisphosphoglycerate not just binds, it's supposed to bind the tetramer, and it's beautifully binding at the active site and blocking this. So basically, you see here, it serves as a lid, and it's the 35 nanomolar. It's beautiful. You see 1,3 bisphosphoglycerate, it doesn't bind effectively. 2,3, it's also not that good. But then this thing is the one that binds. So it has physiological relevance. 2,3 bisphosphoglycerate. If you bind, there are 2 forms that I've shown. I've not gone into the details here. I didn't want to go into it. Here you see ADP. This is the aperture that comes out. The phosphate is here. As the cross that comes out, it attacks. That's what leads to ATP formation. So here, the hemoglobin tetramer relaxed. I said there are 2 conformations. The test when the periods work. Here, xanthine oxidase, Ellsworth et al., wonderful researcher. She showed that hemoglobin with xanthine oxidase once again give high amount of ATP. So xanthine oxidase is a source of superoxide. So it's like that. So basically, if otherwise, if it doesn't find it there, the chances that superoxide and superoxide meet are higher, and then superoxide and superoxide meet, they give peroxide, and that you can't do it. Peroxide is useless. So the paradigm shift here in this field in electrophysiology and RBC chemistry is that ion pumping to ion differentiation. If deterministic ion pumps were to be, why should consumption affect patients? Should sodium consumption affect patients with hypertension? NK is a simple protein, no molecular motor. There is nothing in NK structure that shows that it goes through all these complex 12 cycle, all that. It's a porous, rather loose, open protein. So my proposition is that it is a simple equilibrating mechanism. It's a coarse kind of a filter which recruits superoxide, which enables the equilibration where sodium is lesser here. There is no real stoichiometric outcome there. That is what. And tetrameric. Why do we have tetrameric hemoglobin? Why myoglobin it is monomeric? Because in my muscle cells you have enough mitochondria. You don't need it. Right? So that's the reason. Thank you so much for your patient hearing. So this, I think you need a break. It's 2 hours and 15 minutes already now. And so any question that I had to take, I can take. Yeah, please.
Dr. Murali, I just dive in a minute or two.
No, Professor Matlakonda, please. There is a student asking me a question. We will give you just 1 second. She's asking in the middle. You could not listen
Yeah.
because she's asking a question. She's asking a
Yeah.
question. I'll get to you. Please 1 second. Yeah.
Yeah,
Please give me a second.
go ahead. Go ahead.
Thank you. Yeah. Can you come here? Otherwise, Professor Matlakonda can also hear. Come here and ask so that everyone can hear.
There are other pumps which sometimes act on which we traditionally know as proton gradient. Suppose metal pumps, okay, importers or exporters, which will specifically import or export. Suppose your nickel or cobalt pumps.
There are specific.
Yes, they are very specific.
Definitely.
And suppose that they act on proton gradient. There will be a proton gradient, and based on that, they can act like that. And also, there are pumps for larger molecules like drugs, sugars, and all those. So, there again, there are
I would.
types.
Yeah.
Either they will act on proton gradient or there will be ATP involved
Okay.
in the process. But studies have always shown that there are regions which are highly conserved for those particular molecules.
Correct.
And nutritional studies have also shown that it.
Correct.
So studies have shown that everted vesicles, that is basically you fractionate the membrane, and the pump is embedded in that fractionated membrane, and you make an everted vesicle. So experiments with those everted vesicles where the pumps are already embedded in the vesicles, there also this thing has been demonstrated.
Correct.
So I want to know, like you said about sodium potassium pumps, that this could be the hypothesis. So how is there any way that
Very good question. May I know your name, please?
Yes, Anvesha.
Anvesha. Okay.
Yeah.
Anvesha. Your name is good in the sense that Anvesha means to.
Want to know.
Yeah, want that. Yeah, nice research. Yeah, that's good. So Anvesha, now you are talking about very well established rationale for specific binding. I have never negated that. Do you understand what I'm saying? There are very well established cases, certain affinity driven processes. I never have negated affinity. But we are talking about processes which are very fast, so many thousands occurring in a cycle and all that. I don't have any problem wherein specific binding occurs of say nickel or something where there are multiple amino acids to locate. And the differentiation mechanism has to be specific. It has to be specific. Here, the same amino acid, same thing binds. There is no logic. You understand? Now, I do not negate specific binding and isolation mechanisms for specific ions and metals, but I question, except say, for example, in acid, in stomach, there is pH of 2.5 or less than that, right? There is a definite pushing of protons into the lumen by specific paradiddle cells. Nobody negates that. But when we talk about proton gradient in cells or organelles, when you see most of the cellular system, there's hardly any gradient. This gradient thing was sold to us, which was not existing. If you see immediately after Peter Mitchell proposed this gradient thing, Britton Chance, one of the best experimental scientists of 20th century, he demonstrated that there is no real gradient. So I don't know what you're saying. I do not refute the ion sequestration or confining mechanisms. I am saying, okay, they do exist. I am saying that there is a directionality in transport that is primarily partly there can be an affinity, and then partly it has to be with thermodynamic premises. Because, and when genetics are involved, determinism comes in. Partly because the number of molecules, the number of ions that you have to deal with, if you try to want to determine the concentration and then have specific receptors and transporters, all that, I'm saying for glucose, we know there is. If there are so many molecules, we know there is. And the examples you cited, yes, there is. But on a general level, larger scheme is what I'm talking. Did you get what I'm saying? The larger scheme. I'm not talking about specific instances. there are some drugs that are specifically bound by transmembrane. We target GPCRs, right? I just said. We know that there are receptor binding, affinity binding based on that. But to say that there is deterministic ionic bidirectional pumping by a simple protein like sodium potassium ATPase, that requires too much of intelligence, too much of sophistication, too much of determinism is what I'm saying. In the example that you quoted, I have no problem. I clearly said that at the outside, right? Right? I clearly say that I am not against deterministic affinity-based mechanism. What I am talking about is complementing of those mechanisms by simple thermodynamic rationale that does not necessarily require a very strong affinity-driven logic. You talk about affinity. This process is what you're talking about. This process will be slow, no? This process will be slow. In the sense, kind of slow. From the environment, it will specifically identify an ion and it will specifically take and enable the.
Specific. The reason is also that suppose if I'm talking about the chemical is highly toxic,
Yes.
It is very important that you maintain the homeostasis inside the cell.
Yeah.
Therefore, these pumps are also super specific because that way, because it is needed.
Is the term used as pump or is it some?
It's transporter. It's an
yeah. So let us use transporter because that word is a much better word. Yeah. Pump. That word was pump. When you use pump, the word pump, it somehow is. You understand it? It makes me a little unhappy. Transporter. I say fantastic. We do need transporter.
Another thing, the structural part where you are arguing about lysine and arginine, charged amino acids, and how this Murburn and these two can correlate. So there, I just have one question. This is just I'm trying to visualize and trying to understand how it is. So usually we know that whatever protein is embedded in the membrane, there are regions. The membrane embedded regions are usually very hydrophobic. All the hydrophobic.
Correct.
Are there.
Yes, correct.
And the hydrophilic ones are usually exposed.
Yes.
I'm not saying all of them.
Correct.
Yeah. So, like for those, if you say that lysine, arginine, and this thing, that you are saying that this, so you are saying that this Murburn thing is happening. You are saying it is diffused in the membrane. This membrane is this lipid environment where it is very liquid and it is inside, happening inside.
Yes.
And same, you're saying that this charged amino acids will take part in that.
Yes.
So that is what I'm trying to.
Oh, very good. See, it's a very simple idea. See, this part is in the lipid. This part is in the membrane. This part is above. So what's your question? The question is, here is lipid, the liquid. Here is the lipid. Here is the water. Liquid, water, lipid, water. So here you need to have a mechanism to localize in around this region ROS, reactive oxygen species. Why? See, this is a very porous protein. It is not like it's a breathing protein. It's like you're allowing some interaction with all the components, but then there is a confining of something there. You have a higher amount of probability of having a ROS there, superoxide there. And that's why you need all this high amount of lysine and arginine there. The structure is very beautiful. It's a very simple structure. But how else will I explain the high propensity of it? The point that I'm saying is there is no real reaction occurring there. But the production of superoxide is very essential for that electrophysiology around that region, ion differentiation in that region. So it's not really a conscious pumping or intelligence on the part of that protein molecule. It is a physics-driven, thermodynamically driven, natural, spontaneous process. And that maintains the overall polarity and the nature of the membrane. Did you understand? Here the protein doesn't have any intelligence on its own. The other one is somewhat intelligent. It detects the amount of sodium out inside, and then it pumps 3 sodium out, 2 potassium in, by the same particular locus binding and identify. That is what I have a problem. If you give me a good rationale for specific binding, specific activity, I will like it. I am not a person who does not like determinism. I like determinism. But there are some, besides
Yeah.
deterministic case, there are some.
Can I
Yes.
can I dive in for 1 minute? She has a very good question also. It integrates with your this thing. First of all, the concept that a single molecule is performing a single ion exchange itself is biologically error. Now, sodium is always exchanged in relation to the phosphate. Even if you take the conventional models also, ATPase is associated with phosphate and ADP exchange between the inside mitochondria and outside the mitochondria across the membrane systems. These things have not been given importance because of the stupid theory of Warburg effect. Okay, both Warburg effect and TCA cycle are actually the aberrations of 100 years of aberrations in science. They need to be this thing. There can be a debate, long debate on that thing. I won't go into that particular thing. But any ionic exchange needs to be actually worked out in relationship to the co-transporters of other ions like phosphate ions, right?
For
These
electrons.
ADP ion, phosphate ion, they have to be coordinated along with the sodium and potassium ion. Second important thing is, for those people who have wet labs, I think you people know about Maillard reactions. Glycosylation of proteins, HbA1 is one of the major diagnostic theory, but HbA1 with glucose form has no meaning in that thing. There is a fructated hemoglobin, fructose bound hemoglobin, which alters the position, and this fructose inhibits actually glucose uptake by inhibiting hexokinase in the cells, right? This is one of these things. The shift between actually potassium sodium occurs because of the carboxylations. It suppresses actually the ketohexokinases and then activates carboxylation reactions, oxidative pathways in the cytoplasm. There's a lipid biosynthesis that is taking place, and because these particular ATP citrate and other enzymes have not been identified up to 1950s, and this Warburg effect, which was actually initiated in 1920s, he stuck to that without altering his opinion because of the dogged nature of that man. And otherwise, ATP ATP is a transient molecule. It is not a molecule. We don't have actually physical methods of estimation of ATP. How much of ATP is there? Only we go by sodium, potassium, or magnesium ATPase functions, where hydrolysis is important than actually accumulated ATP inside the cell. We don't have actually data on how much of ATP is there. And then there is also ATP is used only for phosphate exchanges between the organic molecules. The moment actually that function is over, ATP is thrown out in the form of exosomes. There it initiates the myeloid cells, inflammatory cells, by exo ATPase, ENTPD1 are there. There are several isoforms which will be breaking down ATP in the microenvironment, releasing phosphate. That becomes a trigger along with the fructose to remodel the metabolism. These things have to be actually stabilized properly. They are only emerging in the past 5 years.
The thing that the Murburn model says is that the electrophysiology per se, now the presence of superoxide and these kinds of respiratory ions and all that, they kind of affect this solvent lattice, right? And when this process is ongoing, you have a thermodynamic disposition of ion differentiation at all levels. So, and it's on a stochastic chance-based things, and it's not based on deterministic ion pumping based things. Did you understand the difference that I'm saying here? Did you understand the difference? It's not at a single level, protein level thing. It's a phase level kind of a thing. So that only can explain why you say if you have a blood pressure problem, don't take excess salt. So if you take excess salt, the thermodynamic equilibrium gets perturbed. So already your system has got a problem, doesn't know how to manage the ions properly, then it gets even aggravated. If the sodium potassium pump is what is regulating, why should we have a pump? That logic, did you get it? No.
We have another cellular interaction within the neuron, that is Schwann cell. Schwann cell is the source for actually lipid encasement of the axon, and there is a regular turnover of the fats within the Schwann cell that is not taken into account in the present models. That need to be taken into account.
So you have a small break. 2 minutes break. It's already 11:30. Sir, we'll
Yes.
take a break now. 11 o'clock
Thank
or
you.
the break? 11:30 or yeah.
Okay,
So,
okay,
some snacks. Yeah.
that's fine, that's fine.
We'll put a 5-minute break or something. Yeah. No, thank you. Okay.